I'm Allison Mills. I'm a science writer here at Michigan Tech, and I've got Caryn Heldt here. She studies viruses and is in chemical engineering. Tell me what it's like to get such a big grant from the National Science Foundation.
It is a huge honor to be an NSF Career Award winner. What it means the most to my research is the ability to work on something for 5 years and to really study it and delve into it. A typical grant is 3 years long. And so to have 5 years to really study something means a lot, and I think can be very productive.
I want to get into some of the nitty-gritty that you've been researching. And so because there are such tiny little pieces here, if you could talk us through the viruses and the osmolytes that you work with.
So when a virus is in solution, it is surrounded by these water molecules, and that's what keeps it in solution. When we add osmolytes like to structure water, and so they have a lot of water molecules around them. And what happens is that with viruses being hydrophobic, it's pretty easy to pluck some of these water molecules off of the virus structure and have them attached to the osmolyte. So when you have a lot of osmolytes in solution, if you start removing these waters, then you expose hydrophobic surfaces on the virus, which doesn't want to be exposed to water. And so it ends up making the viruses flocculate. And we have found that this flocculation is specific to viruses, and contaminating proteins in solution are not flocculating. And so that adds to our hypothesis that viruses are more hydrophobic, and also allows us to purify the viruses because we can use these large pore size membranes.
Cool. All right. Awesome. One thing that I thought of, because this was a really fun thing that we had done when I first came into your office and was like, what do you do, Caryn? Was we talked about, the osmolytes and what are osmolytes. So I think that this might be time for our crazy, obscure metaphor, is how to explain an osmolyte.
How do you explain it? Well,
yeah.
I don't have the metaphor. I haven't come up with a metaphor yet, but I can explain what an osmolyte is.
Okay.
So osmolytes are actually made to structure water. They're found a lot in deep sea fish. So deep sea fish are in this high salt, high pressure environment, but their cells still need to have water inside of them to maintain volume. And so their bodies produce these osmolytes. They can be amino acids, sugars. There's also some other types of molecules. And when their body produces them, it allows the cells to bring in water against an osmotic gradient, which is where they get the name osmolyte.
You can learn more about how Caryn uses osmolytes to chase down viruses and protein impurities in vaccines by visiting Unscripted. That's mtu.edu/unscripted.